Take note under central dogma processes of living organism. This includes the following processes: the DNA replication. Okay, so please take note, DNA replication, and also gene expression. Gene expression. All right. Particularly under central dogma processes of living organism, this is the flow of information from the DNA, then towards the RNA, then finally making a protein product. That is the final product whenever the central dogma process happens. Okay. And this has a lot of important events that we will be studying today. Okay, so for this figure, this summarizes the central dogma processes of molecular biology. So as I've mentioned, it will start with DNA replication. It will start with DNA replication. Overview lang to ha. So, ibig sabihin sa DNA replication, the DNA here will serve as the template. It will serve as the template para mabuo yung RNA. Alright? And that process that forms RNA from the DNA template, that is known as transcription process. And then, kapag naman ang RNA, it will serve as a template para mabuo yung final product na protein. That process that we denote is translation. Ito yung third process under central dogma of molecular biology. So as you can see, we have DNA replication followed by transcription. Ano yung nangyayari? In general, sa transcription, formation of RNA from DNA. Then kapag translation, production already of a protein from RNA as the template. Okay? Now, all these three processes, we will give them focus. Particularly for today, we will discuss first the DNA replication. So that next meeting, that will be on Friday, we will discuss the remaining two. Kasi madali na lang yung transcription and translation if you know already the foundation under DNA replication. So, class, we will begin with DNA replication. Ano ba yung mga important events na nangyayari under DNA replication? So, bago that, overview muna. Okay, introduction about DNA replication. So, by the term itself, as the term suggests, replicate, replication, ibig sabihin, we are copying the original. Susulat nyo, copying the original. Diba yan yung ibig sabihin ng term. Term na replicate muna. Replication, replicate. Copying the original material. Now, kapag DNA replication na, DNA replication, ibig sabihin, we are copying here an original DNA strand. Okay? We are forming here a new DNA strand. Right? So, yan yung product dapat kapag DNA replication happens. We are forming a new, bagong DNA strand mula sa original DNA strand. O, ano sabi ni Watson's and Crick model when it comes to DNA? Is it complementary or not? It is complementary or not? Okay, very good. Complementary, di ba? Dapat may complementarity rule yung pag-copy ng strand. So, ibig sabihin, such new DNA strand na mabubuo, it has to be complementary. Complementary to the parent strand or the original strand. So, sulat nyo, complementary, dapat yan to the parent strand, or kapag sinabing parent strand, we are also referring this to the original strand. Okay? Ito lang din yung original strand, or in some other references ng biochemistry, this is also the template. Ito na yung template strand. Okay? Yung original ha, kapag sinabing parent strand, parent DNA strand, yan yung original DNA strand. Okay, and another term that we denote for parent strand is template strand. Yan yung pagkokopyahan para mabuo yung new DNA strand, complementary to that template. So halimbawa, o tanong, ilan yung strand sa DNA? Isa, dalawa, tatlo, apat. Isa, dalawa, tatlo, or apat. Very good. Halimbawa, you have here a DNA strand. Diba? Sabi ni Watson and Crick, double-stranded ang DNA. Okay? Yung double-stranded, when they form their secondary structure, maghe-helix yan. Okay, diba maghe-helix? So halimbawa, imagine nyo na lang na nag-helix yan. And remember, sabi ni Watson and Crick, dapat complementarity rule, di ba? Yung nucleotide that represents that DNA strand mag-complement with each other. So halimbawa, yung sequence ng nucleotide is A, T, C, sige, habaan ko pa, mga apat. Halimbawa, G. Then syempre, yung kabilang strand, kabilang strand is also complement, ka-complement niya. Paano ba yung complementary pairings? Yung A dapat paired with what nucleotide? Very good. T. Then kapag G, C. So yan lang po yung complementarity rule. Dapat alam mo yung pairing. So halimbawa, this is the DNA. Original pa to ha, DNA strand. Kasi double-stranded. No? DS, double-stranded yung DNA. Ngayon, if replication happens, dapat itong dalawang, itong nag-helix, di ba mag-helix yan? Dapat yung each of that strand from a DNA strand maghiwalay. Dapat silang mag-separate. Kasi yung isa dyan, kokopyahan, halimbawa itong first strand, magiging template yan para mabuo yung isang daughter DNA strand. Then yung isa din, kokopyahan din siya. Okay? Magiging template din yan. Okay? Or magiging parent strand para mabuo yung new DNA strand. Yan yung concept kapag DNA replication. So halimbawa, itong first lang yung ipakita ko ha, yung first strand, tingnan natin paano yung DNA replication. So halimbawa, naghiwalay na yan, then yung first strand, ito yung first strand. Then sabi, ano yung sequence dyan? It's A, T, C, G. Okay? And paano ba yung direction? Direction ng strand. Halimbawa, yung first, it is typical, 5 to 3, 3 to 5. Sabi ni Watson. Nakalimutan na ma'am. Paano yung typical first strand? It is 5 to 3. Diba? It is 5 to 3. Okay, typical. Meron din namang 3 to 5, pero the typical first strand sa double-stranded DNA, it's 3 to 5. Then kapag complementary, halimbawa this is 5 to 3, yung ka-complement niya, di ba, is 3 to 5. Kasi anong tawag dito na rule? Is it complementary or anti-parallel nga rule or character? Kapag 5 to 3, 3 to 5, very good. Anti-parallel. Di ba? Yan yung characteristic din ng DNA. So, wag niyong kalimutan yung concept na naaral niyo under lesson 1. Okay? You will still use that na mga knowledge in order to understand the preceding lessons. So, halimbawa, ito yung first strand mula sa parent. Diba? This is a template already. Yan yung pagkokopyahan ng bagong DNA strand. So, halimbawa, yung kulay red, this is already the complementary strand. Ibig sabihin, the product one, yung kokopyahan. Kasi copying, yung mangyayari sa replication, copying, no, of the original of a template strand. Okay, so nahiwalay na yung isa, pwede na yan maging template strand para makabuo tayo ng bagong DNA strand. So let's say yung kulay green, ito na yung ka-complementary, no? Ka-complementary strand niya. So what do you think is the sequence? It is T, A, G, and C. Very good. Yan lang yung ibig sabihin or concept sa DNA replication. Okay, overview lang to ha. Meron pong mangyayari na important events detailed na dapat nating i-discuss. Okay, bago ito matapos yung DNA replication. So for now, just know na DNA replication, the original DNA molecule, it forms initially two new DNA molecules. Bakit dalawa DNA molecules initially? Kasi 'di ba, double-stranded, highlight ko, double-stranded yung original DNA strand. Sabi ko, ang requirement or prerequisite para magsimula yung replication, maghiwalay muna yung dalawang strand. And therefore, meron ka ng isang template na pagkokopyahan, another template. So initially, dalawang new DNA molecules yung mabubuo. And each of that new DNA molecules contains a strand from a parent, 'di ba? Ito yung strand from the parent, then the other strand is already from the new strand, the product of copying, no? The product of copying the template strand. So yan yung concept sa DNA replication. Now, with regards to DNA replication, please take note that the process of DNA replication is semi-conservative. Ano ba ibig sabihin ng semi-conservative? Okay, it states here na yung old strand, okay, it splits apart and acts as a template such that yung magiging daughter DNA molecules, yung new, yung bago na mga DNA molecules, each will have one old strand and the other is a new strand. Okay? So ito lang yung diniscuss ko kanina. So halimbawa, this is the parent to, double helix, no? Double helix, diba? Double helix yan, nag-double helix. So yan yung DNA, parent pa yan. Wala pang nangyaring replication. Okay, diba sabi ni Watson and Crick, ang DNA isang double-stranded helical structure. Okay? Ngayon, ang requirement nga, sabi ko kanina, requirement para mabigyan, no? Masimulan yung replication process, dapat ma-separate yung each strand from the parent double helix. So as you can see, yung kulay blue, itong kulay blue, diba mula yan sa original parent double-stranded molecule. Ngayon, naghiwalay. Ang tawag po natin dyan sa process na paghiwalay ng dalawang strand, okay, ng parent double helix, that is unwinding. That is unwinding, or that is also known as unzipping. So kayo na lang magsulat unzip. I-unzip nyo lang, di ba? Parang sa zipper, kung i-unzip nyo, maghihiwalay, di ba, yung dalawang strand. Okay? So yan yung requirement para replication will start. Okay? So as you can see here, di ba, you have already a single-stranded template. Yan yung template. Yung kulay blue. Kapag template, original or new, it is, very good, the original strand mula sa pinaka-root na DNA molecule. Ngayon, sabi nga kapag DNA replication, we are forming here new, no? New DNA strands. Paano? Dapat yung bagong strand is complementary to the old strand. So yan lang po yung nangyari. Okay? Diyan, although it is not being emphasized, but the pairings there must be complementary with each other. Ngayon, bakit semi-conservative? Paano natin maa-apply yung semi-conservative? Sabi nga, kapag semi-conservative, yung mabubuong strand or daughter strand, it must have an old strand and the other one is a new strand. Meron siyang portion na mula sa parental strand, another portion is mula sa new strand, which is, yan yung character, no? Kapag DNA replication happens, yan yung mabubuong daughter cell, meron at merong semi-conservative property na ma-observe. Okay? So yan, yung labeled na blue, old strand, yung kulay red, new strand. Yan, complementary to the template. Sige, recall natin. DNA replication ulit, you have the two parental strand will separate. Two parent strand will separate and each will make a copy of itself. Not literal talaga na copy as in exact nucleotide being copied, but the complementary copy. So you know already the term, complementary copy. Diba? Yan yung character ng isang DNA molecule. Kahit ilang replication pa yung mangyari, dapat yung ka-copy ay yung the copying process must be complementary, no, to the original or the template strand. So after the first round, ayun na nga, ito, after the first replication process, okay, or cycle, dalawa yung nabuong daughter molecules or daughter DNA molecules. Kasi dalawa yung template na pinagkopyahan. Kung dalawa yung template, di dalawa yung mabubuong daughter DNA strand in which each of that daughter strand, one is from old strand or the template original strand and one is already a new strand. Okay? Now, from that first cycle, pwede din naman mag-proceed yung replication cycle. Okay? Ganun pa rin, para ma-replicate ito, maghiwalay dapat ito, yung dalawang strand. Maghiwalay dapat yan, maghiwalay para replication will happen. Okay? So, ulit, meron ka na namang ilan. What do you think? Ilan yung template strand na mabubuo kapag pinaghiwalay mo yung strands mula sa daughter molecules? Dalawang daughter molecules. Ilang template strand yung mabubuo kapag pinaghiwalay ito, daughter molecules. Very good. 4 na template. Kasi sila na ngayon yung magiging kopyahan. Okay? So, ganun lang yung concept ng DNA replication. Okay? So, as you can see here, ito nga yung semi-conservative model. Okay? You have a parental strand, okay? Parental DNA molecule. And para ma-facilitate yung replication, the strands must be separated first. The parental strand must be separated. Ano yung tawag sa process na yon? Pag ipaghiwalay yung dalawang strand, maging single strand na sila, unwinding, very good, or unzipping. Kaya sa first cycle, first round or first replication cycle, meron tayong dalawang daughter DNA strand. Meron bang semi-conservative property na ma-observe? Yes. Kasi sa unang daughter cell, this is made up of an old strand and a new strand. Yan lang po yung concept ng semi-conservative property. Dapat meron siyang, merong nabuong DNA molecule na each strand, okay, each strand must contain one from old, one from new strand. Okay? Now ngayon, from this first cycle, if replication will proceed, so sa second cycle, meron ka ng 4 na daughter cell. not daughter cell, daughter DNA strands. Okay? Daughter DNA strands, 4. Kasi, yun na nga, sabi niya, di ba, nga kapag hiniwalay ito, 4 na yung template. Siyempre, ang template yung pagkokopyahan. Okay? Kaya makakabuo tayo ng 4. Di ba? Kapag hiniwalay yung kulay blue, siyempre, that is a new. Di ba? That is a new strand here. Yung kulay blue. Kaya, kapag replication will happen, of course, the new, okay, will form a conservative. This time, this is a conservative nga type of daughter strand. But again, although merong nabuong conservative, kapag conservative ha, puro new strand yung bumubuo sa DNA molecule. Okay? O kahit may nabuong mga new strand dyan, meron tayong mabubuong semi-conservative property nga DNA, new DNA molecules. Ayan. Nakuha. So you know already why that forms. Okay? Kapag hiniwalay mo yung kulay red, 'di ba yung kulay red from old or from new strand? Yung kulay red diyan. Itong kulay red. New or old? Very good. Old. O, kung siya yung old, papartneran siya kapag nag-complementary, 'di ba bago 'yon? Okay, bago yung strand kapag complementary. So, meron siyang semi-conservative property. Okay, kapag that is being copied, okay, to form a new DNA molecule. So, yan lang yung semi-conservative model. Kahit magpatuloy pa yan, ilang replication, merong semi-conservative property na ma-observe. Okay, so that's the property, distinct yan sa DNA replication. Excuse me. Alright? Now, that is just an overview ng DNA replication. Now, this time, we will discuss the detailed steps involved under DNA replication. Particularly, we will start with the DNA replication under prokaryotes. Although, almost the same lang naman yung steps ng DNA replication with prokaryotes to that of eukaryotic cell, may mga terms lang ng enzymes yung pinagkaiba. Okay? Now, with regards to the steps involved in DNA replication, please tandaan merong 3 major steps no sa DNA replication. Take note ha, DNA replication yung inaaral natin. Okay? Under DNA replication, we have 3 major steps. So please take note. Number 1, we have initiation. Second, elongation will happen. And lastly, termination. These are the 3 major processes in DNA replication. Now, under each major processes, there are important events na kailangan yung malaman. Under initiation, meron tayong i-discuss na mga steps proceeding to elongation, ganun din until termination. Alright? So, under initiation, these are the following important events that happens. Number 1, identification of origin of replication. Okay? Or, tandaan nyo lang yung hint natin dito for initiation, first step, ORI. Yan lang muna yung tandaan nyo, ORI. Okay? Then the second event under initiation, formation of replication fork. Formation of replication fork. Third step, unwinding. Kapag unwinding, joining or separation. Joining or separation, very good. So, separation of the double-stranded DNA. Okay? So, yan yung last under initiation. Then, the step will proceed. Okay? Under step 4, dyan magbe-begin yung elongation. Okay? Kapag elongation na, step 4 na yan, initiation or beginning of the elongation, beginning of the DNA synthesis. Okay? Kasi sa replication, di ba, we are forming new DNA molecule. Okay? So, dapat alam nyo ano yung goal sa DNA replication. We are forming DNA, new DNA. Okay, so under elongation, step 4, initiation of elongation. Fifth step, the RNA primer is being removed. So please take note kapag naka-encounter kayo ng term na excision, ibig sabihin yan is removal. Okay, removal, excision. And then you have their replacement by DNA, removal of RNA primer, and then it is being replaced by a DNA nucleotide. Then sixth step, formation of replication bubbles. Don't worry, we will discuss each of these events today. Okay. Then the last major step under DNA replication is termination. The same steps sa eukaryotes. Initiation, elongation, termination. Meron lang mga terms mamaya sa eukaryotes na pinagkaiba with prokaryotes. But the steps, it's the same. Okay? So we will start, everybody, to the first major process under DNA replication. And that is initiation. Okay, so under initiation, ano nga ulit yung palatandaan sa first step? Ano yung dapat yung tandaan? Letter O. Ano sabi ko? Very good. ORI. Okay, identification of the origin of replication, then formation of the forks, no? Replication, forks, and also the primer. So RP na lang. Sulat ko RP, replication, forks, and then primers also, and then unwinding. So yan yung i-discuss natin ng mga important events under initiation. Okay? So first step tayo, everybody. Please take note ha. Yung una nating i-discuss is under prokaryotes. Okay? Prokaryotes. Ano ba yung halimbawa ng prokaryotes? Kapag prokaryotes, what do you think? Plants ba? Okay, very good. Bacteria. Okay, bacteria is common, no? Kapag prokaryotes, bacteria. So, halimbawa na bacteria, E. coli. Halimbawa lang. Ang daming bacteria. So, let's specify, for instance, a bacteria, E. coli. Now, question, ang mga bacteria ba merong nucleus? Yes or no? Ang prokaryotes ba merong nucleus? Wala. Very good. Kaya, hindi organized yung mga DNA molecules nila, and ano yung formation ng DNA molecules nila. Although yes, double-stranded but circular. So halimbawa, this is the DNA, double-strand, circular, naka-circular arrangement. Okay, di ba yan yung arrangement ng DNA ng prokaryotes. Okay, circular DNA. Ngayon, under step 1, we have to identify saan magsisimula yung replication. Ngayon, di ba ang DNA, mapa-prokaryote or mapa-eukaryote, ang DNA sobrang haba. Marami yung DNA, sobrang haba pa ng DNA. So, saang portion ng DNA ngayon magsisimula yung replication? Okay? Now, para malaman nyo, or for you to identify the starting point, okay, for you to detect the starting point ng replication, you have to know the ORI, or what we call the origin of replication. Now, what is an ORI? Ano ba yung ORI, or origin of replication? As stated in the slide, this is just a short segment. Short segment of what? Short segment of adenine and thymine nucleotide. Ibig sabihin, sa haba-haba ng DNA structure, meron tayong makikitang short segment na puro adenine, thymine na pairings. Yan yung ORI. Yan yung magiging origin of replication. Okay? And mind you, class, na kung ang force of attraction that is being observed between this Base pairs is what we call hydrogen bond. Ano nga ulit yung tawag kapag nagpatong-patong yung mga uh, nitrogenous base? Ano yung force of attraction kapag nagpatong-patong na sila? Nakalimutan na namin, ma'am. It is, what do you call that? Ang example nga doon is van der Waals and also the hydrophobic interaction. Anong tawag doon? That is very important for the stabilization ng structure ng DNA. It starts with letter B. Okay, very good. It's the base stacking. Naalala yung base stacking interactions? Base stacking interactions. Okay? Kapag nagpatong-patong na yan, yung mga base pairings, yung force of attraction in between that piled pairs is the base stacking interaction. Ngayon, when a DNA molecule on that portion na merong 80, 80, 80, puro 80 segments, the base stacking interaction that being observed there is weak, okay, or lesser yung base stacking interaction. Weak or lesser yung base stacking interaction. Now, question, kapag weak or lesser yung interaction being observed there, is it more stable or less stable? Do you think it is more stable or less stable? Very good. So, directly proportional, magiging less stable yung portion na yon. Therefore, kung less stable yan, madali lang yan magiba. Tama? Madali lang yan ma-separate. In other words, no? Madali lang yang ma-separate. That is why, yung ORI sa haba-haba ng DNA, yung portion na yan, yung magiging simula or origin ng replication kasi the AT rich segment there has a less stable interaction and that facilitates melting. That facilitates easy separation. Okay? Now, ano yung protein that is responsible for the melting of that AT segment sa ORI? Ang responsible na protein, okay, for the melting of that AT-rich segment or the ORI portion, that is what we call the DNA A protein. Okay, that is how it is being written. DNA, then capital A, protein. Para saan ulit siya? Melting or separation of the AT-rich segment, okay, in a DNA or ng ORI. Okay, so halimbawa, ito yung portion, itong highlighted with color purple from the DNA strand, nandiyan yung AT-AT segment. So yan yung portion na kung saan magsisimula yung replication. Okay? So yan yung first step. Okay, yan yung first step. So halimbawa, this is a more organized na drawing. So halimbawa, this is a DNA, circular DNA of a prokaryote. Kasi yung prokaryote, circular yung motion ng arrangement ng, although double-stranded pa rin, double-stranded, naka-circular lang sila in the cell of the bacteria. Okay, so sabi nga, halimbawa, itong portion na ito, okay, yung being highlighted or labeled, that is the ORI. So dyan, aatake yung anong protein? DNA, A protein. Kasi nga yung portion na yan, madali lang ma-melt, madali lang ma-separate. Kasi less base stacking interactions are being observed. Okay? Now, ano ngayon yung pinagkaiba ng ORI for the prokaryotic cell versus that of eukaryotic cell? Now, please take note, class, nga for prokaryotes, halimbawa, E. coli, bacteria, the ORI, okay, is only one. Okay? Isa lang po yung ORI. Sa haba-haba ng DNA for that bacteria, isa lang yung makikitang ORI. Whereas, kapag eukaryotic, sa halimbawa sa humans, yung origin of replication, multiple, as being illustrated. Okay, so please take note, multiple ori kapag eukaryotic cell. Not cell, eukaryotic na DNA strand. Okay, DNA molecule. Okay? So yan lang yung pagkakaiba when it comes to the number of ori for eukaryotic versus prokaryotic cell. Ngayon, balikan natin yung action ng DNA protein. Sineseparate niya, no? Sineseparate niya yung portion na 80, puro 80, 80. Yung ori nga part is-separate niya, kaya lolobo. Okay, as you can see, please neglect first the color red. Imagine niyo walang kulay red pa dyan. Yung naghiwalay ha, focus muna tayo sa kulay blue kasi yung original template or the parent double-stranded template is color blue. So halimbawa, after the action of the DNA protein, okay, naghiwalay yung double-stranded. Diba ganito yung double-stranded kanina? O, tingnan ngayon yung kulay blue dito na portion, yung kulay blue lang ha, imagine niyo wala pang kulay red. Kasi ang ginawa kasi sa illustration na to, nag-assume na naman complementary strand na. No, ayaw mabura. Okay, ito na lang. Yung portion dito na kulay blue, yung portion dito na kulay blue, di ba single strand na sila, naghiwalay. Mula double strand, naghiwalay yung 80 portion. 80 portion pa yan ha, nag-melt lang, nag-melt, naglobo, it forms a bubble. Right? It forms a bubble. So yan yung ginagawa ng DNA protein. Okay, sa portion ng ori. Now, as you observed, okay, upon separation, no, of the ori portion, meron tayong makikitang boundary. Highlight ko ng kulay orange. Itong boundary na to. Okay? Kulay orange. Now, yung boundary na yan, okay, being labeled sa taas, okay, it is being labeled, di ba, with a line sa taas, that is what we call replication forks. Okay? Di ba na-mention sa second step under initiation, RP, yung R dyan, formation of replication forks. Ang replication forks, please take note, that is the boundary between the double-stranded DNA strands, okay, between the double-stranded DNA and the single-stranded DNA. So yung pumapagitna dyan, yan yung replication fork. Boundary siya kung saan naghiwalay yung double-stranded into single-stranded. Okay, so yan yung replication fork. Kuha. So yan yung step 2. No? Yan yung step 2. So halimbawa, ito lang din yon. Halimbawa lang ha, hypothetical. 'Di ba mula sa double-stranded, 'di ba, from that, if ito yung portion na may 80, 80, 80, gagawin ng DNA protein, imemelt niya, kaya lolobo. Imemelt niya, kaya maghihiwalay yung double strand. So, erase ko ha. Oh, ayaw na may erase for a while. Ayan. Okay. So, kapag lumobo na yan, automatic, may mabubuong forks. May mabubuong forks. And ito yung forks. Okay? Now, question, in every ori, ilan yung replication forks na mabubuo? Ilan yung replication forks na mabubuo? Very good. Dalawa. No, dalawa. And again, that is the boundary ha. Yan yung boundary between the double-stranded pa, and yung single-stranded. Okay, yan yung single strand. Nako ha. Okay. Now, yung replication fork na yan, that will become the actual site of DNA copying. Ibig sabihin, yung mga succeeding events sa DNA replication, dyan magsisimula sa replication fork. Kasi dyan unang naghiwalay, no, nagmelt yung initiate or beginning portion ng replication. So that will serve as the actual site of DNA copying. Yan yung magsisilbing reference point, kumbaga, ng succeeding or proceeding na mga events under replication. Nakuha? Now, syempre, kapag replication will happen, replication, di ba sa replication we are forming new DNA molecules, And dapat from this template, kapag naghiwalay na yan, ito ay isang template na, ito din ay isang template. Okay? Now, ngayon, since meron pang portion na double-stranded, kailangan pa nating ibuka itong portion na to, highlighted with color red. O, itong portion na to. Kasi, hindi po mangyayari yung replication sa portion na yan kung double-stranded pa lang sila. Remember, ang requirement natin for replication, dapat ma-separate, diba, into single-stranded. Kaya, after formation of replication forks, we have the action of helicases. Diyan na papasok sa story yung helicases. Ano ba yung helicases? Remember, in a replication process, dapat ma-separate yung double-stranded into single-stranded. Ano nga ulit yung term tawag natin kapag separation of the double strands? Separation of the double strand. Letter U. Very good. Unwinding. Yan na yung, very good. Yan na yung unwinding. Okay, yan na yung unwinding. Diyan papasok yung helicases kasi ang helicases na enzyme, they are the ones that unwinds the double helix. Nakuha? So, with regards to the helicases action, okay, etong mga arrow na to, na hinighlight ko, etong mga arrow na yan represents the action or the direction of action ng helicases. Ibig sabihin, bi-directional yung action ng helicase. Kasi, nandoon sa magkabilaang side, ang double-stranded pa. Yan yung ibubuka ni helicases. Kasi, purpose niya, unwinding. Hindi na i-unwind itong nasa gitna. Kasi na-melt na siya, nasugdan na siya melt. So yung gagawin niya, since meron na tayong actual site starting point for the DNA replication, yung gagawin niya, mas ipapalobo pa niya or ihihiwalay pa niya yung natirang double strand. Yan yung gagawin ng helicases, unwinding. Nakuha yung third step. Now from that unwinding, okay, halimbawa, ito yung helicase as being labeled, yung kulay green. Okay? Diba yung direction niya? Isa lang yung fork na pinakita. Isa lang yung fork na pinakita. Itong nasa right. Parang ito lang ba yung pinakita niya dito. Okay? Now ngayon, kapag yung direction or kapag yung helicase nag-move, okay, nag-move siya para bumuka yung portion na to ng DNA, okay, na ay risk na pwedeng mangyari with regards to this single-stranded. Diba? Single-stranded na tong part na to. Ang risk po na pwedeng mangyari is that this single strand may tend to rejoin. They may tend to rejoin, okay, sa double helix. Okay? Imbes na nahiwalay na sila, meron silang tendency or potential na mag-rejoin back. Okay? And remember, para ma-facilitate yung replication, dapat yun single strand. Kasi single strand lang yung template strand. Okay? So yun. Now, para maiwasan, ma-prevent, or to keep that single strand separated, to keep them separated, we have these proteins, no, okay, that enables them to maintain their single-stranded property, the single-stranded binding protein, or this is labeled as SSBP. Okay, the single-stranded binding protein, yung gagawin nila, ito yun sila, kakapit sila, no? Kakapit sila sa single strand para hindi na sila bumalik mag-wind ulit or mag-rejoin, okay, sa double strand, into double strand. Kaya nakalagay dito, di ba? During replication with the fork, the helix destabilizing protein binds to the single-stranded region para ma-prevent yung rejoining. Okay? So yan yung risk with helicases. While binubuka niya yung mga double strand, yung mga single strand dito may potential na mag-rejoin. Okay? Mabalik sila into double strand. So para maiwasan yun, we have these proteins na kakapit dyan. Okay? The single-stranded binding protein. Nakuha? Itong portion na to, review lang to. Okay? Review lang sa mga proteins that was mentioned. Okay? So, na-mention ko kanina, di ba? Yung DNA A protein. Para saan yung DNA protein? Melting or unwinding. Melting of 80 segments or unwinding. Okay, very good. The melting, no, of the ori. It's the melting of the ori or the separation of the ori, focusing on the ori. What about the single-stranded binding proteins? Diba? Ito yung kakapit sa mga single-stranded para hindi na siya mag-rejoin. While the helicases is further unwinding the double helix. While ga-unwind, ga-separate further sa double strand helix yung helicases, kakapit, no, yung single-stranded binding protein sa mga single strand to keep them separated. So review lang to. Okay, review lang yan. Kuha. Okay, next. Ito din, it has something to do with the functions of the proteins mentioned. Meron lang tong figures. Okay, so ulit lang. Kapag na-detect na yung ori, di ba, yung origin of replication, doon mag-a-act yung DNA protein. Ano yung gagawin niya? I-melt niya yon, yung portion na yon. And yun na nga, syempre magfo-form ng bubbles. Magfo-form ng bubbles upon melting. Ngayon, yung single-stranded binding protein, SSBP, kakapitan niya yung single strands para they will be kept separated. Okay? And then, ano yung enzyme na I am being encircled? Ano yung enzyme na to? Ito. This is, kung ito yung DNA A protein, the single-stranded binding protein, what is this? Nga papunta sa mga double-stranded para ihiwalay niya yung mga double-stranded. Okay, very good. It's helicase. No, helicase. Kasi 'di ba, bidirectional yung helicases. Doon siya, ibubuka niya yung mga double strand pa. No? Ibubuka niya yung double strand para ma-facilitate yung replication. Okay? So that will be the end under initiation process. 'Di ba? Sa initiation hanggang unwinding. 'Di ba? Hanggang unwinding. 'Di ba? Ato ang not mnemonics but mga palatandaan is O for ori, the replication fork, and the unwinding. So natapos yung initiation. Okay? Now, ano yung kasunod na major process after initiation? Ano yung major process kasunod ng initiation? Very good. Elongation. Now, bago natin i-discuss yung elongation, let me remind you na habang itong helicases na to, okay, binubuka or sineseparate niya yung double strand into single-stranded, merong risk na mangyayari, okay, sa mga distal part, no, distal portion ng double-stranded DNA. And ano yung risk of that unwinding caused by helicases? Pwedeng magkaroon ng super coiling. Now, para maintindihan nyo yung analogy, bakit hinihiwalay nga ni helicase yung double strand, pero bakit yung nangyayari sa dulo, sa distal part, nagsusupercoil. Okay, para maintindihan nyo itong analogy na to, imagine nyo yung rope, yung tali, yung rope na double strand. Di ba kapag hiniwalay nyo yung rope, okay, although dito sa unahan, yes, mahihiwalay nyo initially, but ano yung mangyayari sa mga distal portion? Di ba? It will magbubuhol-buhol. Did you understand? May I see the chat box if you understand, yes or no? Na-imagine nyo ba yung pagbuhol-buhol sa distal portion, di ba? Ganyan yung nangyayari hypothetically kapag rope, kapag inalogize natin yung DNA strand into a rope. Halimbawa, yung rope is a DNA double strand. Tapos, kapag hiniwalay natin siya, magbuhol-buhol yung mga portion na to. Yan yung supercoiling na nangyayari. Yan yung risk sa unwinding ni helicase. Kaya, ang mangyayari ngayon, kapag may mga supercoils kasi, yes, pwede niyang ma-unwind dito, ma-unwind, pero pagdating dito sa supercoil, ma-stop yung unwinding. Pagdating dito, ma-stop yung unwinding. So, hindi continuous ngayon yung replication. Kaya, good thing naman is meron tayong enzyme that facilitates prevention ng supercoiling. Before that, kung ito ay rope nga analogy, ito na yung sa DNA. Diba? Halimbawa, yung kulay yellow dyan, yan yung helicase. Diba? Sabi ko kanina, yung helicase, siya yung naghihiwalay sa double strand. So once this helicase moves, tingnan yung pangalawang slide, nag-move na siya. Upon moving, ano yung nangyari sa mga distal portion? Nagsusupercoil. Yan yung risk with the unwinding property ng helicases. Ngayon, kapag may supercoil nga, hindi siya appropriate na mangyari continuously ang replication. Kaya, good thing we have enzyme. Ayan. Nawala na. We have enzyme that prevents the supercoiling. And that enzyme is what we call DNA Topoisomerase. Okay, so as mentioned, it is the one that solves the problem caused by the tension generated by the unwinding or winding of the DNA. Okay, yung mga supercoils na nangyayari, yan ay dahil sa tension. Yan ay dahil sa sobrang force na na-exert. Kaya magbubuhol talaga yung nasa dulo. Ngayon, ang gagawin nga ni topoisomerase, pipigilan niya. Now, how is the mechanism ng topoisomerase para mapigilan niya or masulbad niya ang supercoiling na nangyayari? Take note, the DNA topoisomerase has two enzymatic action. It has two enzymatic action. Una, it has the ability to cut. Okay? It has the ability to cut a strand. Okay? A strand or strands. Okay? And that action of topoisomerase is held by its nuclease activity. So yan yung unang enzymatic action ng DNA topoisomerase. Second, yes, kinat niya nga, pero temporary lang yon. Okay? Temporary lang yung pag-cut niya kasi kapag na-relieve na yung coil, ibabalik niya, i-reseal niya ulit yung kinat niya. Yan yung second enzymatic action ng topoisomerase, and that is being held by ligase activity. Kapag naka-encounter kayo ng ligase enzyme, yung yung hint lang dito or key term for ligase is to join. Okay, to join. To join, ligase yan or ligation. Kaya reseal yung equivalent activity niya. Nakuha? So, ulit, yung DNA topoisomerase is the one that relieve the supercoiling. Kailangang i-relieve yung supercoiling. Okay? Bakit? Para continuous yung replication process. So, kailangan natin siyang i-relieve. Paano nare-relieve ni topoisomerase? Yung supercoiling. Una, via its nuclease activity, ikakat niya, okay, yung supercoil temporarily. Ikakat niya temporarily, and kapag na-relieve na yung supercoil, i-reseal niya pabalik, no? Redundant, no? Reseal is sealing back naman. So, i-reseal niya yung kinat niya initially, okay, para mabuo ulit yung double strand. Nakuha? Yan naman yung ligase activity niya. Okay, so meron siyang nuclease activity, cutting, ligase activity, resealing, or joining again. Okay, the segmented nga strand. Now, with regards to the nuclease activity niya, let me highlight the nuclease activity niya. And again, it has something to do with the cutting property niya sa strand. There are two classification ng topoisomerase based on its nuclease action, yung cutting action niya. Madali lang to ma-memorize kasi pag sinabing topoisomerase 1, ibig sabihin, it can only cut one strand of the DNA double helix. So as you can see, isa lang yung nakat niya na strand. Whereas, kapag topoisomerase 2, it cuts both strands of DNA double helix. Yan lang yung palatandaan natin sa dalawang types ng topoisomerase. Kapag topoisomerase 1, isa lang yung kaya niyang i-cut. Kapag topoisomerase 2, yung dalawang strand, yung kaya niyang i-cut temporarily. Take note ha, temporary lang to na action kasi yung gagawin niya at the end, okay, kapag na-relieve na yung supercoil, it will rejoin, no? It will rejoin the initially broken nga strands or cutted nga strands. Okay, so yan yung action ng topoisomerase. Nakuha? Okay, so that ends the initiation process. Okay, sige. So before tayo ulit mag-proceed sa elongation, let's have a recap first. Okay, recap muna. You answer ha, my question. Kapag original strand, is it new or template strand? Original, is it new or a template strand? Very good. It is a template strand. Ano yung tawag natin sa process of separation, the double-stranded? Separation of the double-stranded. Very good. Unwinding or unzipping. Kapag yung nabuong strand, merong property nga, ang isa from old strand, yung isa has a new strand. Ano yung tawag sa property na yon? Very good. Semi-conservative. Now, what do you call the origin of replication? Ano yung origin of replication natin? origin of replication? That is 3 letters only. Very good. That is ori. What about the protein that is responsible for keeping the strand single-stranded? For keeping the two-strand single-stranded. You can have the abbreviation. Yes, it is the single-stranded binding protein. What about the enzyme responsible for unwinding? Responsible for unwinding. Very good, helicase. Ano naman yung enzyme responsible for melting the ori? Melting the ori. Okay, very good. DNA, a protein. O ilan yung ori sa prokaryotes? Okay, very good. Isa. Kapag eukaryotes, multiple. Okay, very good. Multiple. Sige. So, let's continue. So, I think, klaro na sa tanan yung initiation. Now, let us have elongation this time. So, before na ako i-discuss yung nasa slides provided to you, mag-pen and board muna tayo. Okay? Para mas maintindihan yung nasa slides. Okay? Sige. So, first thing first, with regards to elongation, dito na natin pahahabain, no? Pahahabain or magkakaroon ng DNA synthesis. Okay? Kasi yung ending ng initiation is unwinding, di ba? So nabuka na, maseseparate na, may mga single strand na, dito na magsisimula yung DNA synthesis, elongation. Now, question muna, sabi ni Watsons and Crick, ilan ulit yung strand ng DNA? Ilan yung strand ng DNA? 2. Okay, with regards to the complementary rule, paano yung pairing ng bases? Paano yung pairing ng bases? Paano yung pairing ng bases? G tsaka A ba? C tsaka T ba? Okay, that is for DNA. Very good, ha? That is for DNA. A is paired with T, G at saka C. O paano naman kapag RNA? Question, kapag RNA. Very good. A paired with U, then ganun lang din sa G. Okay, paired with C. So please take note, ha, kapag RNA na gani, it's A paired with U. Kasi yung thymine, it is replaced with uracil kapag RNA. Okay. Now, with regards to elongation, dito na natin ma-observe yung complementary formation of what we call complementary DNA strand. Okay? O kapag sinabing complementary, is this a new strand or the original strand? Is this the new strand or the original strand? Very good. New. Okay, naalala, kapag nagbuo na tayo ng new DNA strand, dapat complementary siya to the template strand. So imagine niyo, ha, please take note, complementary dito, product na tayo dito sa product na side, yung pagkopya na natin. Okay, from the template. Now, with regards to elongation, the enzyme that is responsible, that facilitates elongation, okay, or the synthesis of DNA, that is the enzyme DNA polymerase. Okay, so via the term itself, polymerase, di ba pag polymer, it has something to do with polymerization. Pagpapalaki, pagpapahaba ng molecule. So DNA polymerization, that is responsible for elongation process, synthesis of complementary DNA strand. Now, with regards to DNA polymerase, please take note na hindi siya basta-basta nagfa-function or nag-act forming the complementary DNA strand. Hindi po siya agad-agad bumubuo ng complementary DNA strand. Maarte yung DNA polymerase. Dapat naasa siya makita ay siya mo function. Okay? And yung pre-requirement niya, okay, take note, the prerequisite or the pre-requirement niya para siya ay maging functional into the process of elongation, dapat meron muna siyang ma-detect na RNA primer. Okay? Meron dapat siyang ma-detect na RNA primer doon sa pagkokopyahan. Okay? Now, ano ba yung RNA primer? Ano ba yung RNA primer? Ang RNA primer, this is just a very short segment of RNA. RNA nga, no? RNA. Very short ng RNA. Approximately, it only contains 10 nucleotides, no? 10 nucleotides. So, take note, that is an RNA, ha? RNA. So, dapat meron munang ma-detect si DNA polymerase na 10 nucleotides RNA made. RNA made. Okay? Yung tanong, paano ba nabubuo yung RNA primer? Yan yung tanong, 'di ba? Kung saan lugar ni siya pag-form ang RNA primer? Para kung ma-form na siya, elongation will happen via polymerase. Now, the enzyme naman, dito naman tayo, paano na-form itong RNA primer? Ang enzyme naman that is responsible for the formation of RNA primer is what we call primase. So, okay lang ang term kasi primer yung mabubuo via primase. So, sounds alike. Okay? So, kapag primase na enzyme, primer yung binubuo niya, the RNA primer in particular. Nakuha? Now, let us discuss now the character of this primase. How does it form the RNA primer? Kasi kailangan muna natin malaman paano mabuo yung primer para kapag andyan na siya, elongation will follow, no? Via polymerase. Now, ngayon, ano yung character ng primase? Ano yung character ng primase? The primase is an enzyme. Please take note that it reads the template. Okay? It is able to read the template from 3' to 5' direction. Yan yung way niya para magbasa ng template. Question, ano yung template? Original or the new strand? Is it the original or the new strand? Template. Template strand. Ano ba talaga? Okay, very good. It's the original. Di ba sabi ko, yung template, okay, yung template, yung unang slide na diniscuss natin, the parent strand is also known as the template strand or the original strand. Okay, so orig. Paano siya nagbabasa ng template? From 3' to 5' direction. Okay, so para mas maintindihan, let's have an illustration. So let's say for example, this is the replication fork. Okay, so nabuo na yung, ito, may nagbubbles na, ayan. Ayan. So this portion, di ba, is the replication fork, replication fork. So unang replication fork, second replication fork. Now let me magnify only one. Okay, let us magnify only one lang ha. Only one lang sa. Kasi if you understand the concept, okay, kahit sa isang fork lang, ganun din yung mangyayari sa kabilang fork. Okay? So let me magnify this part, yung kulay green. So halimbawa, itong kulay green, magnified na siya. Pinalaking fork, no? Pinalaking replication fork. Okay? And 'di ba sabi ni Watsons and Crick, ang DNA strand anti-parallel yung direction. So halimbawa lang, okay, halimbawa this first strand is 3' to 5'. Halimbawa lang ha. Okay, pwede din naman 5' to 3'. Okay? Now, and then the second, since anti-parallel, ano yung magiging direction sa baba? Sige, you type. Kung anti-parallel, very good. So this will be 5' to 3'. Okay, so minagnify lang natin ha, minagnify lang natin itong part na to. Ngayon, this is the replication fork, okay? The replication fork, the boundary, no? The boundary that separates the double strand to the single strand. Ako ha? Now, sabi dito sa primase, ang primase daw ay kailangan para mabuo yung RNA primer. Paano tayo makakabuo ng primer? Si primase, yung gagawin niya, it will read the template from 3' to 5'. So halimbawa, this is the first template. Okay? Tagsa man gyud. Isa ra man gyud ang template na pagkuula, ngano na nga YouTube naman. Wait sa ha, ayan. Okay. Then ito, this is another template. So una tayo sa isang template. So isa-isa lang. So okay. So we have the first template yung nasa taas muna. Okay? Babasahin daw ni primase from 3 to 5. Example, yung reference point natin is the replication fork. Replication fork. So kung 3 to 5 yung basa ni primase, paano yung direction ng action ni primase? Is it, ito yung reference point ha, replication fork. Is it away from the fork or towards that fork? Is it away or towards the replication fork? Paano niya babasahin? Sabi dito, 3 to 5. And then the template has the direction already of 3 to 5. So paano? Very good. It is away from the replication fork. Kasi ito, sabi ko, di ba, ito yung reference point. Okay? Kung yan yung replication fork, di ba, dyan yung actual site? Kasi dyan muna bumuka eh. Di ba, dyan muna bumuka yung single strand. Dili ka pwede mag-replicate kung double strand pa. Di ba, akong gibalik-balik ganina, kung naka-double strand pa, walay replication mahitabo dira nga part. So dito ang mangyayaring replication is dito na portion sa single strand na. Okay, so let me erase. Bago yung elongation na mangyari, dapat meron ngang RNA primer. Nabubuo siya with primase. So ibig sabihin, yung basa ni primase sa taas na template is away from the replication fork. So dito magsisimula, let's say color yellow yung primase. Paano ba? Ayan. Color yellow yung primase. Diyan siya magsisimula. Kaya kapag nagbuo siya ng ka-complementary strand in an RNA form, kasi RNA primer, di ba RNA? Primer is an RNA daw. Yung 10 nucleotides, dito magsisimula. Nakuha. Let's say, color violet na lang. Color violet. O yan. So with that, meron na tayong RNA primer on that portion. O ganun din yung mangyayari sa kabilang template. Diba? Meron din tayong template dito. Paano babasahin ni primase yung template from 3 to 5'. O ngayon, kung from 3 to 5' yung basa niya sa template, ano ngayon yung direction kung ang reference point is the replication fork? Is it towards that replication fork or away from the replication fork? Very good. This time, towards, no? Towards the replication fork. Okay? Towards the replication fork. So, ang point of or starting point ni primase is, halimbawa, dito. Nakuha? Halimbawa, dito. So, ito yung, halimbawa, 10 nucleotides. nucleotides. Ito yung mga primer. Okay, this is the primer, the RNA primer, then the RNA primer. Okay? So, yan yung purpose ni primase. Okay, so magdepende yun siya sa direction ng template na kinokopyahan niya. Kung ito, ang template, di ba, originally sa second template is 5 to 3 yung direction, and yung basa ni primase is from 3 to 5, dito talaga siya magsisimula kasi yung basa niya is from 3 to 5. Unlike yung nasa taas, naka 3 to 5 direction na, so same direction, no? Same direction with the template, yung pagbasa niya, forming the complementary na strand. Okay? RNA ang mabubuo dyan. RNA, initially, yung mabubuo. So you know already what I mean kapag nag-base pairing yan sila. Kapag RNA primer, ito yung magiging pairings. Okay, ito yung magiging pairings. And how many nucleotides sa RNA primer? Itong sa violet, how many nucleotides sa RNA primer? 10. Very good. Okay? So ngayon, meron ng RNA primer na nabuo. Meron ng RNA primer na nabuo. Therefore, DNA polymerase will now be activated. Okay? DNA polymerase will now be activated, and it is the DNA polymerase that facilitates elongation. Yung magpapatuloy dito, magpapatuloy ng complementary strand is already the DNA polymerase. The DNA polymerase, yung magpapatuloy. And you know already what I mean sa complementary strand. Sa first slide pa lang, di ba, gihatagan tamuog, overview what is replication all about. From the template strand, kokopyahin. Dapat complementary. Okay? Yung new strand. Ka-complementary niya ng new strand yung old or the template strand. Okay? So dito tayo ngayon sa DNA polymerase. So no problem already with the RNA primer. It is being formed via the primase. Paano niya binabasa yung template? 3 to 5 direction. 3 to 5 direction. Okay? So let's have now the function of the DNA polymerase. So let me draw again. Okay, same lang, kulay green ulit ha. Ayan. So drawing ko ulit, ganun din, yun lang din nga fork. Okay? So sabi dito yung direction, di ba? Is 3' to 5', 5' to 3'. Okay? Then, ayan. Then, meron na tayong nabuo na primer. Yan lang man yung gustong makita ni DNA polymerase. Dapat may primer siyang makita. Okay? So, dito, if this is the replication fork, okay, meron ng primer dito. 10 nucleotides. Dito din, merong primer. Okay, yung kulay violet, RNA primer yan. RNA primer, short segment only. Short segment only. Okay, of a complementary strand. Ngayon, ipasok natin yung DNA polymerase enzyme. Ano yung kinakabit ng DNA polymerase? Siyempre, DNA, so mga DNA nucleotide. Okay? And you know already what is the pairing involved kapag DNA. It should be A is paired with T, G is paired with C. Nakuha? Kapag RNA kapag RNA, ito lang yung pinagkaiba. A is paired with U, uracil. Okay. Now, paano ngayon yung reading ni DNA polymerase? Kung ang primase 3 to 5, then sabi ko kanina, si DNA polymerase, siya yung magtutuloy sa elongation. Ano ngayon yung basa ni DNA polymerase sa template? Is it the same or opposite? Is it still the same or opposite? Siya man daw ang magtutuloy. Siya ang magtutuloy sa sinimulan ni primase. Very good. The same lang. Kasi itutuloy niya lang. Nasimula na ni RNA, itutuloy niya lang. Kasi short lang naman yan. Hindi pa yan DNA strand na buo. no? Hindi pa yan DNA strand na buo. Kasi RNA, di ba to? Made up of RNA. This is made up, yung primer, made up of RNA yan. So, hindi pa yan buong DNA molecule talaga. Marami pa pong mangyayari. Okay? So, DNA polymerase, the same with primase, it reads the template, okay, from 3' to 5'. Then, kasi itutuloy niya lang yung sinimulan. So, halimbawa, kakabit dito, let's say, yung kulay red. O, ayan. Dito kakabit, no, yung enzyme na polymerase. Kakabit dyan yung enzyme nga polymerase. Kasi may na-detect na siyang RNA primer. Di ba yan lang yung kailangan niya? Pag may RNA primer, okay, mag-a-act na ako. Ipagpapatuloy ko yung sinimula ni RNA primer. Okay? That's why strands will form. Okay? Ipagpapatuloy niya din yung sa kabila, strands will be formed. And since magkaiba yung direction nila, ang isa away from the fork, yung isa towards the fork, tama? Towards ito sa fork. Ngayon, dalawa yung posibleng strand na pwedeng mabuo with DNA polymerase. Upon reading the template from 3 to 5, forming the complementary DNA strand, dalawa yung strands, complementary strands nga pwedeng mabuo. Yun na nga, yung isa is away, yung isa is towards what? Towards the replication fork. Okay? Now, with regards to the specific names of this complementary strand na mabubuo, please take note nga kapag away from the replication fork, okay, so itong strand na mabubuo natin dito, that is known as lagging strand. Okay, that is known as lagging strand. Or this is also known as Okazaki. Okazaki fragments. Fragments. Kayo na lang magpatuloy. Fragments. Okay? And then, kapag naman yung strand, complementary strand na nabuo towards the replication fork, that is termed as leading strand. Okay? Leading strand. Okay? Leading strand is also known as continuous strand. Continuous strand. Lagging strand is discontinuous strand. Okazaki fragments also. Lagging, putol-putol. Leading, continuous, walang putol. Okay? Sige, let's try to understand bakit tinawag na leading strand kapag continuous. Okay, let's start with ito, yung towards. So, ito yung nasa baba, di ba? So, ibig sabihin, yung mabubuo nating strand dyan upon the action of this DNA polymerase, isang leading strand. Continuous strand yung mabubuo. Bakit? Continuous strand. Okay, please refer to the, excuse me, to the direction. Okay? 'Di ba yung direction is toward the replication fork? Okay? Now, what do you call that enzyme that unwinds the remaining double-stranded DNA? Ano yung tawag natin sa enzyme that unwinds the remaining double-stranded DNA? Kasi dapat nating ibuka pa, 'di ba? Yung mga remaining double strand. Ano yun ulit? Yung enzyme na yun? It starts with letter H. Very good. Helicase. Kung ito yung direction, nandito yung portion na double strand, the direction of this helicase is of course to the left. Kasi nandito pa yung mga double stranded. Para magpatuloy yung replication on this portion, 'di ba? Dapat ihiwalay ni helicases, okay? Yung naka-double strand into single strand. Now, bakit leading strand is continuous? Kasi upon forming the complementary strand, let's have this color. Yan, kulay blue. Upon forming the complementary strand, di ba, towards the replication fork, kapag bumuka na yan, itong portion na to, kapag bumuka, magpapatuloy lang. Tama? Magpapatuloy lang yung pagbuo or elongation. No? Magpapatuloy lang yung elongation via the DNA polymerase. Kasi towards the replication fork, yung direction niya. Okay? Upon reading, di ba? The 3' to 5'. Yan yung reading niya sa template. Sinusunod niya lang yung RNA primer. Nakuha? That's why continuous. Hindi putol-putol yung replication. Okay? Kumbaga, same yung direction ni helicase with the polymerase. Kaya continuous. Papunta sa left yung helicase para bumuka yung remaining double strand, magiging single strand. Ganon din yung direction ni polymerase on that template strand, okay, towards the replication fork. That's why we have what we call leading strand or continuous strand. Comparing sa taas, ano yung nangyayari sa taas? Is it the same ba yung direction ni helicase towards the polymerase? The same or opposite? Helicase papunta sa left. Yung polymerase sa taas, away. When it reads the template from 3' to 5'. 3' to 5'. So, away from the replication fork. What do you think is the direction? Opposite ba sila ng direction ng helicase and then the polymerase? Yes, obviously, very obvious, opposite, sobrang opposite. Okay? Please take note ha, malalaman nyo kung towards ba or away if you know the reading. Dapat, alalahanin nyo yung paano mag-read yung enzyme. Paano siya nagbabasa ng template. 3' to 5'. Yan yung reading niya. So, titingnan nyo yung template. Ano yung arrangement ng template? Then, yung reference point nyo is the replication fork. So, away siya sa replication fork, opposite with the direction of the helicase. Habang, kung i-imagine nyo, habang si polymerase, kinulayan ko ng red, habang si polymerase papunta siya dito, forming the complementary strand, tapos si helicase binubuka niya yung double-stranded part dito, magiging single-stranded, di ba dito? Kasi ibubuka man, ihihiwalay, i-unwind ni helicase. Unwinding will cause separation of the double strand. Kapag naging single strand niya, kailangan na yan i-replicate, no? Kapag may helicase, sineparate niya yung double strand, single strand na yan, dapat i-replicate din yan. Kaya, habang papunta dito si polymerase away from the replication fork, madedetect niya na, meron na namang single strand na naiwan ko sa likod. Kaya, magbubuo na naman siya sa likod ng RNA primer, polymerase action ulit. Then, magpapatuloy ulit si helicase, madedetect na naman niya na, sa likod ko may naiwan na naman na single strand kasi binuka ni helicase, babalikan niya yung part na yon, bubuo na ulit siya ng RNA primer ulit, then tsaka yung action ng DNA polymerase. So, if you will try to imagine, putol-putol yung mangyayaring replication dito. Putol-putol yung mangyayaring strand or mabubuong strand. Kaya, lagging strand yung tawag. Nakuha ba? Lagging strand yung tawag or discontinuous. Okay? Can you type in if you understand the difference between leading strand and lagging strand? Do you understand leading and lagging? Yes or no? Okay, so yes. At least meron akong nakikitang yes. Okay.
So be honest na nga, I know you're still analyzing it. As long as you understand, i-recap na lang ninyo siya. As long as you understand the concept, you just have to read again your notes and i-recap ninyo. So yan yung kaibahan ha, excuse me, ng lagging o Okazaki fragments and the leading strand. Nakuha? Sige. Ayan. Okay, so if we go back to the slide, go back tayo sa slide ha. Ayan. Ito yung sinasabi kong step 4, the initiation of elongation. Kailangan muna may RNA primer. 'Di ba? Na-discuss ko kanina sa pen and board. Dapat meron munang primer. O ilan ulit yung nucleotide kapag RNA primer? 10. Very good. How does it read the template? How does it read the template? Anong direction? 5 to 3, 3 to 5. Okay, very good. 3 prime to 5 prime. Okay, very good. Ano yung enzyme that forms RNA primer? Ano yung enzyme? Very good. Primase primer. Okay, very good. Now, kapag may primase na, mabubuo yung RNA primer. Kapag na-detect na ni polymerase na merong RNA primer sa complementary strand, ipapagpatuloy niya yung elongation, forming the complementary DNA strand. Okay? Now, question. Kapag primase and polymerase, the same ba yung direction? Primase tsaka polymerase, same ba yung direction? Same ba yung direction nila? Yes or no? Yes. 'Di ba yung primer 3 prime to 5 prime, it reads the template. Polymerase, di ba, ang polymerase, dudugtungan niya lang, ipapagpatuloy niya lang yung RNA primer. So 3 prime to 5 prime lang din. That's how it reads the template. That's how it reads the template. So same direction sila ng primase tsaka polymerase. Okay? Now kapag Okazaki fragments, continuous, discontinuous strand. Okazaki, discontinuous. Kapag leading strand, continuous or discontinuous? Okay, very good. So you understand already. So ito lang din yung sinabi ko about RNA primer. Ito lang din yung diniscuss ko. Again, hindi po magkakaroon ng DNA polymerization, hindi mag-a-act yung DNA polymerase kung walang RNA primer. Okay? Kaya kailangan ng RNA, this is just a very short piece of RNA base pairing. Okay? To the DNA template. Ilan ulit? 10 base pairs. Okay? Ito lang din yon, the enzyme responsible for the synthesis of RNA primer is the primase. Okay? And ano sabi natin? Anti-parallel siya. Anti-parallel siya sa DNA template. Ito lang din yung diniscuss ko. Yung unang strand lang is naka 5 to 3 reading, not reading, direction. And the complementary template is 3 prime to 5 prime. Ngayon, if 5 prime to 3 prime ang reading, not the reading, the direction of the template, yung kulay blue ha, kung ang kulay blue nga template, okay, 5 prime to 3 prime, yung direction, if a primase acts to it, 'di ba? Babasahin niya into 3 to 5 prime. Kaya andito yung kulay green, yan yung RNA primer. Nandiyan yung RNA primer. Ito lang din yung diniscuss ko ha sa pen and board. Nakuha? Nakabaliktad lang yung presentation ko. Depende yan sa how it is presented, yung template strand. Okay, pwede mag-una yung 3 prime to 5 prime or the other template is 3 prime to 5 prime. And then when it reads the primase, when it reads the template, primase, dapat 3 to 5 prime. Kaya andito nabuo yung primer. And then for the other strand, titingnan niyo yung primer, andito. Kasi nga, 3 prime to 5 prime, it reads the template. Okay? Ngayon, yung nasa taas na template, upon forming the or upon elongation, no, upon elongation via the polymerase, DNA polymerase, continuous strand yung mabubuo. Let's highlight it with color red. Continuous. Bakit continuous or leading strand yung mabubuo? Kasi towards, yung direction nila is towards the replication fork, same direction with the helicase kung ibubuka ito ng helicase. Helicase. Same direction with the helicase. Continuous leading strand yung mabubuo. Unlike yung nasa baba, okay, yung nasa baba, di ba, this is the beginning, the RNA prime. So, away from the replication fork, yung direction ng pagbuo ng complementary strand. Kaya, habang binubuka ni helicase, etong remaining double strand, maiiwan sila na single-stranded. Kaya, bubuo na naman ulit ng RNA primer dyan na parte ang primase, and then tsaka mag-a-act yung polymerase. Lagging strand yung mabubuo, discontinuous Okazaki fragment. Okay? So, yan lang yung na-discuss ko kanina. Then, ulit lang, DNA polymerase, same yung reading niya with the primase. 3 to 5 prime direction. The reading, how does it read? Okay? The template, same with the primase. Kasi, ipagpapatuloy niya lang yung primase, yung ginawa ni primase sa RNA primer, 3 to 5 direction. Then yung na-synthesize, of course, na DNA, syempre, this is a complementary already. This is a new, ito, di ba? This is already a new DNA strand. Di ba? The primase muna. The primase, as stated sa taas, it reads the template 3' to 5'. So, walang problema dito sa reading the template. Okay tayo dyan. Bakit yung mabubuong complementary strand is already a 5' to 3' direction? Kasi, as you can see, it reads the template from 3' to 5'. Diba kapag Watson's and Crick nga model of a DNA, dapat yung unang strand, kung ni-read niya as 3' to 5', dapat yung ka-complementary niya would be 5' to 3'. Kaya yung mabubuong complementary strand, kapag pinagpatuloy yan ni DNA polymerase, 5' to 3'. Complementary, anti-parallel. Okay? Anti-parallel dapat yung direction. Diba this is the rule or the model ng DNA. Kahit i-replicate mo pa yan, ilang replication, dapat anti-parallel to each other yung strand. O ganon din dito. Sa baba, binasa niya from 3' to 5'. So dito yung arrow towards the fork. Tama? O kung 3' to 5' yung pagbasa niya, yung mabubuong strand dyan would be 5' to 3'. Nakuha ba? O, now, you type in if you understand. Anti-parallel lang, anti-parallel rule. Okay, very good. O, yes. Ano lang, anti-parallel rule lang. Binasa niya ng 3 to 5, di yung magiging ka-complement niya, 5 to 3, yung direction. Yan lang yung ibig sabihin sa slide na yon. Okay? So, ayan. Then again, it is responsible for elongation. It is responsible for elongation, yung DNA polymerase. Sige. O, ito lang din to reinforce what we have discussed sa earlier nga mga slides. Ibang ano lang siya, orientation. 'Di ba? Yung mga example ko kanina is etong fork na to. So ito, yung kabilang fork naman yung ibang orientation ng replication fork naman yung ginawang example. So ganun pa rin naman, yung template strand. Halimbawa, yung template strand is 3 to 5, yung nasa taas. Siyempre, yung mabubuong strand would become 5 to 3. Okay? Yung reading. O, dito naman, from 3, dito yung 3, ito yung 3 to 5. Okay? That's the second template, which is anti-parallel to the first template. Kasi 3 to 5 dito, 5 to 3. Now, how does it read? How does the primase read the template? 3 to 5. Kaya, dito yung starting point, then away from the fork. Okay? Kaya yung mabubuong strand, opposite sa pag-read niya. Opposite sa pag-read niya. Binasa niya ng 3 to 5, yung mabubuong strand would be 5 to 3. 5 to 3. Lagging strand ha, lagging strand. Kapag away from the fork, if towards the fork, continuous, or leading strand. Ayan. So, yun lang yun, yung diniscuss ko kanina. Ito lang din, paulit-ulit siya. 2 types of strands na nabubuo with DNA polymerase. Ito na nga, yung leading strand and the lagging strand. Kapag towards the replication fork, advancing means towards the replication. So, continuous ang mabubuong complementary DNA strand. Kapag naman lagging strand, the direction being copied, okay, sa strand is away from the replication fork. So, away from the replication fork, discontinuous yung strand na mabubuo. Or this is also known as Okazaki fragments.
Still under DNA polymerases, in particular, nasa major process pa rin tayo under elongation. Now, with regards to DNA polymerases, they have two important properties. First, as mentioned earlier, we have polymerase action, very important for elongation. So ito lang yung na-discuss natin kanina na property of polymerases or DNA polymerases. Yung kanina na-discuss natin na with elongation, the enzyme responsible for the formation of complementary DNA strand is the polymerase, the DNA polymerases. Now, when we try to connect this to their enzymatic action, the elongation property of the DNA polymerase, that is due to its polymerase, to be specific. Polymerase enzymatic action. So please tandaan, kapag elongation property ng DNA polymerases, that is due to the enzymatic action na polymerase activity niya. Polymerase activity. Now, another distinct property of DNA polymerases is they have proofreading activity or property. Pag sinabing proofreading, ibig sabihin, they can double check what they did. Diba? Upon elongation, they are placing or forming a new product, a complementary DNA nucleotide from the template strand. So upon placing a DNA, a complementary DNA nucleotide, babalikan niya or i-double check niya yung nilagay niya. Okay? Kung tama ba or mali. Yan yung proofreading. That is why we could really say na yung DNA replication or yung pagbuo ng bagong complementary DNA strands, that is strictly regulated, that is being controlled. Kasi kapag hindi yan strictly regulated, pwedeng magkaroon ng mutation, mali yung pag-pair up ng mga base pairs, then that will lead to diseases, that will cause diseases. So please take note, another distinct property of DNA polymerases, proofreading. They could double check what they did. Okay, upon elongation. Now, kapag naman i-connect natin ito, i-relate natin sa enzymatic action ng DNA polymerase, the proofreading action nila, it's because of its exonuclease activity. I repeat, it's because of the exonuclease activity, kaya merong proofreading action or property yung mga DNA polymerases. Now, Still under DNA polymerases, this time let us talk about the two types, particularly of DNA polymerases. Simulan natin sa unang type. There are actually different, no, different types of DNA polymerases. So what I have discussed earlier in a slide is the general properties na meron ang mga types of DNA polymerases. Now, first type, okay, that we will be discussing under DNA polymerase, by the way, with the types of DNA polymerases na i-discuss ko, still prokaryotic ha, types of DNA polymerase under prokaryotic cell. Karyotic. Okay. So first type na i-discuss natin is DNA polymerase 3. DNA polymerase 3. Now, one of the characteristic of DNA polymerase 3 is elongation. So please take note, it has elongation activity or property. So ito lang din po yung na-discuss natin na type of polymerase that is very important for the elongation. Okay? Or yung pagbuo ng complementary DNA strand sa product. Diba? Kapag elongation, we are talking about here new strand. I have mentioned earlier that the polymerase enzyme, it has the characteristics, or it reads the template from 3' to 5'. Okay, sige, recall natin. Okay, so halimbawa, this is a template. Ayan, template strand. So pag sinabing template strand, ito ay galing sa old strand or original DNA strand. So halimbawa, we have here the original strand, and then the original strand, yung template strand will have the direction of 3' to 5', and it has the nucleotide DNA na let's say T, C, and G. Now, I have mentioned earlier that the polymerase action or character is that it is capable to read the template. It reads the template from 3' to 5'. Kaya, upon elongation, halimbawa, this is already the new, no? Dito magaganap yung elongation. When it reads the template, dito siya magsisimula. Kasi ito yung 3, di ba? Ito yung 3'. So, it will place a DNA nucleotide, kasi we're talking about the polymerase, DNA polymerase already. So, halimbawa lang, mayroon ng RNA primer, etc. So, doon na tayo mag-focus sa elongation portion. Diba? Elongation will happen with polymerase enzyme. So, yung A, it will complement it, or maglalagay siya ng ka-complement na DNA nucleotide, which is T. So, ayan. And then, A, then followed by G. So, you know already why G yung nilagay ko dyan. Then dito, C. Kasi standard base pairing. Okay, so tandaan niyo yung standard base pair. Ngayon, upon elongation, since we're talking now about the new product, this is already the new strand. Tama? This is a new strand. This is the old. This one is the old strand. Okay? So, ito yung nangyayaring elongation. Elongation, yung pagbuo ng complementary DNA strand. Now, as you can see, yung formation ng product is mula dito ang
right. Kasi nga it reads that template from 3' to 5'. Yan yung na-discuss natin na reading capacity ng polymerase. Ngayon, with regards to its polymerase product na nabuo, polymerase activity, okay, yung magiging direction dito, itong strand na to, itong highlighted with color red, yung magiging direction niya, syempre, hindi 3' to 5'. Kasi dapat Watson's and Crick, yung mabubuong ka-complementary strand, it should be anti-parallel with each other. Yung strands, yung dalawang strands, okay, in forming the DNA, it should be anti-parallel. Kung yung isa, 3 to 5, yung the other strand would be, ayan, 5' to 3'. That is why, with the polymerase, DNA polymerase 3, yung elongation activity niya, okay, is because of its 5', okay, to 3' polymerase action. Okay, so kumbaga, yung meron siyang 5' to 3' polymerase activity in elongation. Okay? So, yan yung unang character ng DNA polymerase 3, elongation. Meron siyang character na elongation because of its 5' to 3' polymerase activity. Okay? So, etong activity na to it will depend sa product. Okay? Etong new product na nabuo. Alright? So, ayan. Now, another property naman or character ng DNA polymerase 3 aside from elongation. Okay? So, second we have, sige, lagay ko dito letter A. Then, dito naman letter B. We have proof reading. Proof reading. I've mentioned earlier na yung proofreading, ibig sabihin, double checking of what they did during elongation. Ano ba yung ginagawa during elongation? Naglalagay siya ng ka-complement DNA nucleotide. Okay? Ka-complement DNA nucleotide. Now, ngayon, during proofreading, okay, nangyayari ito, halimbawa, etong dinro ko, this is, halimbawa this is the polymerase 3. Okay, this is polymerase 3. Siyempre, ang polymerase 3, siya yung nag-fill in. Nag-fill in ng ka-complement DNA nucleotide. Okay? Base sa template strand. So, T yung nilagay niya. Now, iiwanan niya yung T, okay? Then mag-fill in siya sa kasunod na gap. Okay? Mag-fill in siya sa next gap para ma-elongate. Okay? Ma-elongate yun. So, prior to placing a complementary nga base, okay, or DNA nucleotide, bago siya maglalagay dito halimbawa ng A, bago siya maglalagay complement ng thymine, okay, babalikan niya, okay, yung T. Babalikan niya, lilingon siya, okay, on the left. Okay, lilingon siya on the left. So, left. Okay, lilingon siya on the left kasi nga proofreading. Titingnan niya kung tama ba yung nilagay niya. Tama ba yung pinair up niya sa A. Now, in the event, halimbawa, hypothetical, C yung
Paglingon niya doon, kasi meron siyang proofreading activity. Paglingon niya, para ma-double check, uy, C yung nalagay ko. Ipo-proofread niya. I-correct niya yung mali niyang pinlace na pair. Kaya, i-correct niya, maglalagay siya ng T. So, yan yung proofreading. So, kung titingnan natin, yung direction niya kapag proofreading to the, sa halimbawa ha, dito sa slide na to, di ba? Opposite sa polymerization direction. Di ba ito yung polymerization direction or the elongation. Polymer, that is the DNA synthesis or the elongation. So, kung to the right, yung polymerization, yung ginagawa sa proofreading, opposite ng direction ng polymerization. Kasi nga titingnan niya, babalikan niya yung nagawa niya. Okay? Okay, upon elongation. So, this time, kapag proofreading, yung direction would be 3' to 5'. And what is the enzymatic action that we could relate DNA polymerase action kapag proofreading? I have mentioned it earlier, di ba? Kapag proofreading, that is because of its exonuclease activity. So this time, kapag proofreading naman na characteristic of DNA polymerase 3, that is due to its 3' to 5' exonuclease activity. Okay, so yan yung proofreading action niya ng anong type ng DNA polymerase 3. Okay, sige, so recap natin. Once again, we have first type of DNA polymerases for prokaryotic cell. We have the DNA polymerase 3, meron siyang elongation characteristic. Yung elongation property niya, it's because of its 5' to 3' polymerase activity. Kasi, bakit polymerase? Kasi nga, yan yung related, di ba, na enzymatic action for elongation property ng polymerases. Bakit 5' to 3'? It is because of the formation of the new product upon elongation. If it reads, di ba, if ang polymerase capable siya mag-read ng template from 3' to 5', syempre yung mabubuong new product, okay, always 5' to 3'. Okay, kahit baliktarin natin yan. Okay, baliktarin natin yung template ha. Kahit yung template, if it is represented from 5' to 3', remember na yung polymerase, halimbawa this is the polymerase. Again, ang polymerase, yung activity niya is yung reading activity niya sa template para mag-elongate, babasahin niya yung template from 3 to 5'. Kaya dito pa rin siya magsisimula, no? 3' to 5'. Yan yung mangyayaring elongation. Okay? Kaya yung mabubuong new strand or complementary DNA strand upon elongation, it's 5' to 3' pa rin. No? 5' to 3' pa rin. Kasi yung direction is to the left. Okay, so yan yung tandaan nyo ha, with the elongation characteristic ng polymerases. Halimbawa, polymerase 3, 5' to 3' polymerase. So, product tayo. Okay, titingnan nyo yung product. Then, for the proofreading, so yun na nga, upon placing, okay, a complementary DNA strand, bago siya mag-place ulit ng ka-complement sa pangalawa, lilingon siya, okay, lilingon siya sa kabila, titingnan niya, i-double check niya on the opposite side ng polymerization, yung kung tama ba or mali yung nilagay niya. Kaya 3' to 5' kasi opposite. Mula dito, That's part of 3 prime, no? Then going to the 5 prime direction. Kaya 3 prime to 5 prime, and what is the enzymatic action related to proofreading? Exonuclease. Okay? So please take note of that. So that's the first type of DNA polymerase. Ayan. So for this slide, ayan. So this is again one of the property ng DNA polymerase 3. Ano sabi kanina, kapag DNA polymerase 3, then elongation, uh, it is because of its 5 prime to 3 prime polymerase action. Okay? Kapag elongation, it has something to do with a 5 prime to 3 prime direction. Kasi nga, yung mabubuong daughter strand, okay, or yung bagong strand, it should be anti-parallel, no? Anti-parallel dapat sa parenteral strand. Babasahin niya yung parenteral strand, di ba? Ng 3 prime to 5 prime. So, yung mabubuong daughter strand, kung ni-read niya from 3 prime to 5 prime, the opposite, anti-parallel dapat yung daughter strand. Kaya 5 prime to 3 prime polymerase action kapag elongation character ng DNA polymerase 3. And then, yun na nga, yung proofreading naman, okay, as being stated, it is the double checking of nucleotide after it is being added, or particularly the nucleotide is the DNA kasi we're talking about DNA replication here. Okay, so with regards to proofreading, this is mediated by the 3 prime to 5 prime exonuclease activity of the polymerase 3. Okay? So kapag exonuclease, nagpo-proofreading siya. Okay? Kapag mali yung nilagay niya, then it will remove that nucleotide. Okay? What is the direction? Yun na nga, opposite to the polymerization or elongation direction. So halimbawa, if we try to check this figure on the right, halimbawa, although it is not clear, but I think medyo klaro naman siya. So halimbawa, you have a polymerase 3. No? A polymerase 3, and then yung kulay blue dito, ito yung old strand. Kapag old strand, yan yung template strand or original strand. Okay? And then yung kulay orange, ito na yung new. So again ha, yung new, dyan na yung elongation. Okay? Kapag elongation, we are forming new complementary DNA strand there. Okay? So, as you can see, yung direction, 5 prime to 3 prime. Okay? Now, kung titingnan natin yung highlighted with color green, itong part na to, ito. Diba? Yung from the original strand, T, it is being paired with C. And then, iniwan niya yung C dito. Then, bago siya nag-place dito sa, bago siya nag-place ng ka-complement DNA nucleotide for the G, nilingon niya yung C, and napagtanto niya na, uy, mali yung nilagay ko. Kasi, di ba dapat, kapag T ang nasa template strand, it should be paired up with A. A with T, and then yung G, it should be with C. So, dapat A yung nilagay. Okay? So, upon proofreading, nakita niya na mali yung nalagay niya, ipo-proofread niya yon with, paano yung proofreading? That is, because of its 3 prime to 5 prime activity. Kaya mula dito, diba, this portion is the 3 prime. So, bago siya mag-place ng ka-complement with G, babalikan niya yung C for 3 to 5 prime direction. Nakuha? And that is related to exonuclease enzymatic action. Kaya, upon removal, okay, remove niya, at the end, it will incorporate now the correct nucleotide, which is Okay, so yan yung concept ng proofreading, double checking of the DNA nucleotide after it is being added. Kaya very strictly controlled, regulated yung ng DNA replication. Okay. Next, another type of DNA polymerase is the DNA polymerase 1. With regards to DNA polymerase 1, this is the one responsible for excision of RNA primer. Kapag sinabing excision, this is synonymous to the term removal. So, ibig sabihin, itong type na enzyme, DNA polymerase 1, is responsible for removal of RNA primer. Now, if we recall, RNA primer, this is very important para mag-function yung polymerase. Naalala, yung initiation ng elongation process, hindi po magfa-function ng basta-basta ang polymerase kung wala siyang na-detect na RNA primer. So, kailangan i-synthesize muna yung RNA primer via a primase. And paano nagre-read ng template yung RNA primer? Di ba? It reads the template. Halimbawa, this is the template. Okay. Di ba? And the template, halimbawa, is 3' to 5' yung direction. This is the old. Okay. Template old, ibig sabihin the original or the template strand. Okay. Then, halimbawa, itong red, yan, that is the RNA primer. Okay, that portion, itong portion na to is RNA primer. Ilan ba yung nucleotide under RNA primer? Diba? It is just a short segment. Okay, para ma-initiate yung elongation process. And there are about 10, diba? 10 RNA nucleotides, okay, to form RNA primer. Now, kapag RNA primer, this is part somehow of the new strand or daughter strand and should be in 5' to 3'. Kasi it reads the template from 3 to 5. So, dapat anti-parallel. Anti-parallel based on Watson's and Crick. So, halimbawa lang ha, hypothetical. I'll place example of RNA nucleotides. Let's say U, and then you have A, and then you have C or G, and then U. So, halimbawa lang. And there are 10. Okay, continue lang yan. Then 10, about 10, so that consists of an RNA primer. Alright? Now, yung ginagawa ng DNA polymerase 1 with regards to removal of RNA primer, yung direction niya would be based on the direction of the product itself. How is the RNA primer being arranged, diba? RNA primer is being arranged from 5' to 3'. Therefore, kung i-remove niya yung mga RNA nucleotide mula 5' to 3' din. Nakuha? So, i-remove niya individually yung RNA nucleotides. So, halimbawa, let me remove niya yung U. Okay, so yan yung ginagawa ng DNA polymerase. Siyempre, upon removal ng U, ano yung gagawin niya? Mag-fill in siya ng gap. Now, with regards to the removal na activity ng polymerase 1, removal of RNA primer, that is because of its 5' to 3' exonuclease activity. So, tandaan ha, that is due to its 5' to 3' exonuclease activity. Bakit 5' to 3'? It's because of how the RNA primer is being arranged. Diba? Parte siya ng daughter strand, and the daughter strand is being arranged from 5' to 3'. So, it will individually remove the RNA nucleotide in a primer from 5' to 3' direction. And the one responsible to that activity ng polymerase 1 is its
5' to 3' exonuclease activity. Okay? So, yan yung dahilan bakit meron siyang activity to remove RNA primer. Once again, due to its activity na 5' to 3' exonucleases. Now, syempre kapag ni-remove niya ngayon yung U, halimbawa kasi individual niya nire-remove. Ni-remove niya yung U, syempre may gap, may blank, no? May space. Hindi mo na siya magpo-proceed sa next. Okay? Mag-fill in siya dyan. If i-filled up niya yung gap via its polymerase activity. And remember, polymerase in general, di ba? Polymerase in general, meron silang polymerase elongation action. Okay? And polymerase activity. And paano yung direction kapag polymerase activity? Siyempre, 5' to 3'. Nakuha? Kasi we're talking about the product here. No? The product, you have to focus on the product. Kapag polymerase action, okay, polymerase characteristic, you have to focus on the product itself, okay, or the new strand already. Okay, so paano ba nabubuo yung new strand? 5' to 3' direction. Yung elongation, di ba, is 5' to 3' direction. Kasi nga, binabasa niya yung template in opposite way. So, dapat anti-parallel yung mabubuong new product. Okay, so nakuha. And syempre, bago siya ulit mag-proceed, okay, in removing again the RNA nucleotide, i-check niya ulit kung tama ba yung nilagay niya. So, halimbawa, in-erase niya to, ayan, ni-replace na niya ng DNA nucleotide. Ayan. Halimbawa, this is an A sa taas. Halimbawa lang ha. Halimbawa, A yung nasa taas. Okay? So, i-replace niya ito ng T. Okay? Kasi, a T base, nitrogenous base is the one that is present, okay, or observed sa DNA. Hindi naman U. Okay? Kapag RNA nucleotide, then that's the time U, yung paired up with A. Okay? So, yun na nga, dahil sa DNA, 5' to 3' polymerase activity ng polymerase 1, na-filled up niya yung gap pagkatapos niyang na-remove yung RNA nucleotide dyan. Then yun na nga, bago siya mag-proceed on removing again this one and then replacing that, bago siya mag-proceed, lilingon siya kung tama ba yung nilagay niya na base pair. Okay? So, dyan papasok yung proofreading activity niya. And kapag proofreading ulit, exonuclease activity yon ng polymerase. And what is the direction kapag proofreading exonuclease activity? Opposite direction ng polymerization. And that is 3' to 5'. Nakuha? Diba? Bago siya mag-proceed dito, babalikan niya. Okay? So, opposite direction from 3' to 5'. Nakuha. So, that's the proofreading activity naman of DNA polymerase 1. Okay. So, review natin na mabilisan. So, you have another type of DNA polymerase 1. This is responsible for the removal of RNA primer. Okay, removal of RNA primer. Now, with regards to its activity na removing RNA primer, that is due to its exonuclease activity, particularly its 5' to 3' exonuclease activity. Then, kapag naman polymerase activity ng DNA polymerase 1, if i-fill up niya yung gap, di ba? Yung gap upon removing the RNA primer, so that is based on its 5' to 3' polymerase activity. And then, yun na nga, i-double check niya yung nilagay niya na DNA nucleotide. And that is via proofreading activity niya,
which is 3' to 5' exonuclease activity. So yan yung other type ng DNA polymerases. Okay? Now this slide here, so review lang din to with regards to the activity ng DNA polymerase 1 on removing the RNA primer. So as you can see, halimbawa, if this is a polymerase 1, if this is polymerase 1 and you have here the lagging strand, diba? Obviously, this is a lagging strand kasi putol-putol. No? So, sa lagging strand, yun na nga, ni-remove yung kulay red, ni-remove niya yung RNA primer. Yan yung main, no, main talaga na activity ng polymerase 1, removal of RNA primer. Kasi magre-replace siya dyan ng DNA nucleotide. Kasi 'di ba, yung discussion natin is about DNA replication. Hindi naman habang buhay na mamamalaan yung RNA primer. Okay? Temporary lang yan para mag-function yung polymerase. Then, there will be a time na i-remove siya para mabuo na completely yung bagong DNA. Okay? Yung bagong DNA. So, ayan. So, as you can see, it removes these RNA primer. Okay? Then, upon removal, syempre, i-replace niya. So, kung titingnan natin yung removal lang, yung removal activity niya, it's from 5' to 3'. Look at the direction, di ba? From 5' to 3'. Kasi base yan sa arrangement ng RNA primer itself. The RNA primer, nabubuo siya from 5' to 3'. Okay? So, yan yung main activity ng DNA polymerase 1. So, ito, review ulit to sa enzymatic activity ng dalawang types ng DNA polymerases. So, balikan natin ulit yung kakadiscuss lang. Yung polymerase 1, again, it has a 3' exonuclease activity. Kapag ito related, kapag 5' to 3' exonuclease activity, this is not the one related to proofreading ha. Kapag 5 to 3, arrangement yan ng RNA primer, so removal of RNA primer. Okay, so this is the one related to excision or removal of RNA primer. Okay, kaya yes, meron si polymerase 1. And dito sa polymerase 3, wala naman tayong na-discuss nga meron siyang activity towards removal of RNA primer. So wala siyang 5 to 3 exonuclease activity. Then the rest, dito na sila magkakaroon ng similarities. Yung polymerase 1 and 3. Both have 5' to 3' polymerase activity. Ito yung related sa letter E, elongation. Okay, nakuha or polymerization. And then, kapag naman 3' to 5' exonuclease activity, ito naman yung related sa letter P, proof reading. Okay? Ayan. And then, this would be the last step under elongation. Okay, so this involves ligation. Ligation of the newly synthesized DNA segments. If you can still remember the Okazaki fragments, di ba yung Okazaki fragments, particularly putol-putol, lagging strand, yung nabubuong new strand or DNA, complementary DNA strand. So, para mabuo or ma-join together yung mga putol-putol na strands, okay, we have this enzyme DNA ligase. So, siya yung nag-join together or responsible para mabuo yung continuity ng strand mula segmented na DNA strand or lagging strand into one strand continuous. Okay, so yan yung importance ng DNA ligase. So overall, that ends the
elongation steps. Steps under elongation. So, recall natin, 'di ba? You have there the initiation of elongation. Dito nagsisimula kapag initiation, meron tayong RNA primer. Importante yung RNA primer para masimulan yung DNA synthesis, masimulan yung elongation. And again, the enzyme responsible in general kapag elongation are the polymerases. Okay, so discussed already the two types of polymerases. You know already what is the type of polymerases that is very much involved to elongation, yung mahabang elongation, and the one responsible for the placement or replacement ng RNA nucleotide into DNA nucleotide. Anong type na yun na polymerase. And then after that, we have the, yun na nga, kapag na-initiate na yung DNA synthesis, you have the removal of the RNA primer. And then finally, there will be dito papasok yung DNA ligase action. Okay, para sa Okazaki fragments. And then lastly, we have the major process termination. So ito na yung mark ng end, no, ending ng processes under DNA replication. So again, I repeat, sa termination, it already marks the end of the DNA replication processes. So it is being mediated by sequence-specific binding of terminus utilization substance to the replication sites, stopping ultimately the movement of the replication fork. So yung concept lang dito sa termination, you just have to take note na kapag yung helicases na action, kapag nag-stop na yung action ng helicases, that would eventually stop the movement of the replication fork. Kaya ngayon, magsa-stop na ngayon yung polymerization process or the DNA elongation. So, yan yung termination process. So, yun lang yun. Okay, so just take note of that. So, review natin yung overall direction ng DNA replication. So, halimbawa, yung kulay orange dito. Ayan, yung kulay orange dyan being labeled, those are the helicases. Ano sabi natin sa helicases? Siya yung responsible, letter U, unwinding or unzipping. Kaya dyan siya nakalagay sa double strand kasi siya yung responsible para maghiwalay yung double-stranded. Okay, yung double-stranded pa. So yan yung helicases. So by the way, yung pinakita lang dito na replication fork, isa lang. Okay? So you have a replication fork. So andyan yung helicases, itong kulay orange. Now, ano naman yung mga labeled with kulay yellow? Yung mga kulay yellow dyan, yan yung single-stranded binding protein. Para saan yon? Yung single-stranded binding protein. They are the ones that keeps, no? It keeps the single-stranded from rejoining the double strand. So, kumbaga, kakapitan nila yung mga single strand para mananatili silang single-stranded. Okay? Kasi ang tendency, meron kasi silang risk na pwede silang mag-rejoin sa double strand. So, we have this single-stranded binding protein to stabilize, no, the unwound nga parenteral DNA. And then, itong kulay red na to, yung putol-putol, nakikita nyo, putol-putol. Anong type ng strand yan na nabuo? That is the lagging strand or the discontinuous strand. Diba? Kaya putol-putol yan. And then yung kulay red dyan, being highlighted, no? Mga kulay red dyan, di ba? RNA primer yan, the RNA primer. And sino yung enzyme responsible para ma-synthesize yung RNA primer? The primase, the RNA primase. Alright, and then you have here, the ligase. The ligases, siya yung nagjo-join together ng mga fragments of the lagging strand. And then yung nasa taas naman, ito yung leading strand. So just take note ha, kailan nabubuo yung leading strand, kailan nabubuo yung lagging strand. Okay? So kung titingnan nyo lang, while looking at this figure, di ba yung lagging strand, yung direction niya, direction ng product is away from the replication fork. So kapag away from the replication fork yung direction ng polymerase, then that will form a lagging strand. Pero kapag yung direction ng polymerase is towards the replication fork, the same with the direction of the helicase, it will form a leading strand or continuous strand. So yun lang yung mga na-discuss ko kanina. Ayan. Then ito din na slide, it also review. Okay, it helps you review the proteins, the enzymes that are involved or that were mentioned earlier along the process ng DNA replication. Diba? We have the DNA polymerases. So I have mentioned in general, kapag general nga, is very important for elongation polymerization. Okay, and dyan na papasok yung two types, no? So it's already your job to scan over the types of DNA polymerase. We have the DNA polymerase 3 and the DNA polymerase 1. So please take note of the difference. Ano yung mga similarities and differences nila when it comes to their enzymatic activity. Then we have helicases responsible for unwinding. Topoisomerase, naalala? Topoisomerase is responsible to relieve the super coiling. Okay, it relieves the super coiling. And meron tayong dalawang klase ng topoisomerase. Topoisomerase 1, topoisomerase 2. So madali lang ma-recall ano yung difference ng 1 and 2. It has something to do with their cutting. Okay, yung cutting nila na activity. If it only cuts one strand, then type 1. If it cuts the two strands ng DNA, then it is topoisomerase type 2. Then we have the primase. Primase, again, responsible for the synthesis of RNA primer. Very importante yung RNA primer to initiate the DNA synthesis. Then we have the single-stranded binding protein para mapigilan yung pag-rejoin ng mga single-stranded into double-stranded. Okay, and then we have the DNA ligase. This is very important to seal the Okazaki fragments on lagging strand. Alright. Now, para mas ma-reinforce natin yung discussion, I will be sharing to you a clip about DNA replication kung talagang naintindihan nyo yung DNA replication process. So let me share to you another screen.
So again, this video, this is just a reinforcement if talaga bang naintindihan yung steps that are involved in the DNA replication, particularly for prokaryotic cell. So I'll play the video and may mga portions nga ako siyang i-pause para ma-relate natin sa discussion ko kanina.
DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The two strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called
Okay, hindi lang na-mention dito, but the initial step under initiation is, di ba, to determine the origin of replication, and that is the ORI. Okay, the ORI. At saka, that will be followed by the unzipping, of course, which is done by the enzyme helicases.
Called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a
Now, I want you to take note the portion kung nasa ang side papunta ang helicases. So, tingnan nyo ha, yung helicases, ina-unzip niya to the left. Ito yung pinapakita dito na representation.
new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the
Alright, so ito yung diniscuss ko kanina with regards to the activity of the polymerase in relation to elongation. It is because of its activity na 5' to 3' polymerase activity. So please take note
The
of
new
that.
strands of DNA, the leading strand, is made continuously. The DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again.
So as you can observe, there is a difference on the formation of the strands between the leading strand and the Okazaki fragments. And it has something to do with the direction ng polymerase from the replication fork. So kung titingnan nyo, magka-opposite direction yung helicase. Di ba yung helicase kanina is to the left. Kung titingnan natin yung polymerase ngayon is to the right or away from the replication fork. Kaya magkakaroon talaga ng segmented DNA. Okay, na mabubuo. Yan yung Okazaki fragment. Kasi habang hinihiwalay ni helicase to the left, yung double strand, merong matitirang single strand. So, i-replicate yun. Kaya, babalikan, magpo-form na naman ng RNA primer, saka naman yung short segment ng DNA nucleotide. So, meron na namang binuka si helicase. So, yun na yan. So, there will be an Okazaki fragments or lagging strand. Repeated siya na process.
Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
So, yan din discuss ko about the conservative property of DNA replication sa preliminary slides, no? Na meron talagang semi-conservative property na ma-observe kahit ilan pang replication na mangyayari, okay, sa DNA. Okay, so we're done with the DNA replication for prokaryotic cells. Now, at this juncture, let us now proceed to the DNA replication naman for eukaryotic cells. So please pakatandaan na yung eukaryotic DNA replication process, it starts, okay, under the S stage ng cell cycle. Okay, yung S stage ng cell cycle, dyan po magaganap yung DNA synthesis. So just take note of that for eukaryotic cell. Kasi under eukaryotic DNA replication, madali lang din naman siyang maintindihan. Almost the same lang yung processes under prokaryotic cell. Only that, may mga terms or may mga enzymes or proteins na madadiscuss natin in addition. Okay? Or may mga simpleng terms na magkaiba sa prokaryotic DNA replication. But when it comes to the overall process, halos parehas lang din naman. Okay? So, halimbawa, for eukaryotic DNA replication, sabi dito, both the DNA parenteral strands, both the DNA strands serves as the template, which is the same with the prokaryotic DNA strand, di ba? So if this is the, di ba, if that is the DNA, paano ba, ano? Okay? Okay, kapag DNA strand from the parenteral, if these are the parenteral strand, kapag in-unwind yan, yung dalawang strand na yan from the original strand or parent strand, yan yung magsisilbing template. Ganon din sa eukaryotic DNA replication and prokaryotic. Another semi-conservative process is observed under eukaryotic DNA replication. The same. No, na-discuss din natin yan under prokaryotic DNA replication. Now, what about the ORIs? Origin of replication. So, dito sila nagkakaiba. There are multiple ORIs sa eukaryotic DNA replication, whereas for prokaryotic, there are only one ORI. And again, it occurs, the DNA replication for eukaryotic cell, it occurs at S stage of the cell cycle. And please take note of this. The mechanism termination under eukaryotic DNA replication is not clear. Okay, it is not clear. It is said to be unclear. Now, with regards to the terms or the proteins that are involved during the processes sa DNA replication, meron lang naman mga konting terms na kailangan yung i-take note. Pero when it comes to their functions, halos the same lang naman with prokaryotic. Okay, prokaryotic DNA replication. Dito lang nagkaiba lang yung terms, magkaiba lang yung terms. But with the function, almost the same. Okay? So yung mga polymerases sa eukaryotic DNA replication, they are being designated with a Greek letter. So kung sa prokaryotic, di ba yung polymerases, we have polymerase 1, then we have polymerase 3. So eukaryotic DNA replication, they are being designated this time not by Roman numeral number but with Greek letters. So halimbawa, polymerase alpha, this is the one responsible for the primase activity. Kapag primase activity, kumbaga, siya yung responsible for the RNA primer synthesis. So it's the polymerase alpha. Then we have the polymerase delta. It is the one that elongates the leading strand. Take note, it elongates. Please take note of the term ha, elongate. Hindi po synthesis, hindi po create, but elongate. So polymerase delta, leading strand siya related. Then polymerase epsilon. Epsilon yung symbol na yan, epsilon. That is responsible naman for the elongation of the lagging strand. And please take note, okay, that both the polymerase delta and polymerase epsilon, they have 3' to 5' exonuclease activity. O, saan to related kapag 3 to 5' exonuclease activity? Is it elongation or proofreading? Letter P, so it's proofreading. So since meron silang activity to elongate, syempre upon elongation, bago sila maglalagay ng complement on the next, lilingon sila sa kabila, titingnan nila, i-double check kung tama ba yung nilagay nila. So 3' to 5' exonuclease activity. Then we have polymerase beta. This is the one that is analogous to the E. coli DNA polymerase 1, okay, which is important for excision of the primers. Okay, or it carry out repair. So yan yung polymerase beta. So these are the terms na kailangan yung malaman or take note kasi ito yung pagkakaiba lang nila sa prokaryotic terminologies, okay, under DNA replication. Now, when it comes to processes naman, almost the same lang. Diba? We also have helicases that unwinds the double-stranded DNA. We also have the single-stranded binding proteins. No? It binds to the single strands para makip yung single-stranded lang. Okay? And then we have the primers are being laid down, okay, para ma-initiate yung DNA synthesis. And then, take note, the synthesis of the leading strand is being carried out by the polymerase delta. And take note, the synthesis of the lagging strand is being carried out by the polymerase alpha. Take note of the terminologies, ha? This one is synthesis. It's not elongate. Okay, synthesis. So ito, summary lang naman to sa eukaryotic, the difference, no? Difference between the DNA replication ng prokaryotic at saka eukaryotic. So dito sila parehas. Semi-conservative is being observed sa DNA replication ng both cells. Primer dependent. Kumbaga, magsisimula, mag-i-initiate yung DNA synthesis kapag merong na-detect na RNA primer yung polymerase. Then both parenteral strands, they are being used as templates. So dun sila nagkakaparehas. Now, saan sila nagkakaiba ulit? Single oris lang sa prokaryotic, multiple oris sa eukaryotic. Then, we have DNA polymerase 3. It creates the leading and the lagging strand. And then, for the synthesis naman of the lagging and the leading strand, we have the polymerase alpha and polymerase delta respectively. Alpha for the lagging, delta for the leading strand. Creation, or the synthesis, not the elongation. And then, we have supercoiling, no, sa prokaryotic. Okay, but for the eukaryotic, supercoiling and there is telomere shortening. Right? So overall, we are done with DNA replication under eukaryotic cell. Okay, so parehas lang talaga, parehas lang yung processes. There are certain terminologies lang na dapat yung i-take note for eukaryotic DNA replication. Now, for the preceding slides, ayan, this involves presentations ng mga DNA-related na mga problems or conditions or diseases. Kumbaga, kapag may alteration sa base pairings or may removal sa nucleotide base in a DNA during DNA replication, then that will cause a disease, no? That will cause a disease. So, madali lang din naman sila maintindihan. You just have to read them all. So, halimbawa, these are certain factors that causes DNA damage. So if you expose to UV light, radiation, any chemicals, extreme chemicals, or other agents, those are certain factors that could lead to DNA damage. No, paano ba yung mechanism ng DNA damage sa katawan? So halimbawa, kapag may base alteration, okay, that will lead to DNA damage. Okay, halimbawa, yung pairings ba? Yung A, it is being paired up with, let's say, C, then so on. Okay, so mali yung base pairs, then that could lead to mutation, then ultimately longer na genetic problems. okay, related to DNA. Then, another mechanism that would lead to DNA damage kapag may removal, na-remove or may gap, okay, sa mismong DNA, replication process or series ng nucleotide base, then that will also lead to DNA damage. But good thing naman is we have different mechanisms for DNA repair. Okay? So, kumbaga, kahit marami pang mga adverse stimuli nga may balak na i-alter, okay, or will cause mutation, may balak na mag-cause ng mutation sa DNA, meron din tayong mga compensatory mechanism naman sa katawan that would somehow repair, okay, the alteration sa DNA. So halimbawa, we have mismatch repair, base excision repair, nucleotide excision repair, and double strand break repair. Madali lang ito maintindihan. Just know what is the problem and ano yung ginagawa, okay, based on their mechanism. So you just have to read this table, okay? Sabotable lang naman siya. Okay, so that these are the 4 mechanisms of DNA repair. But yun na nga, kapag hindi na kinaya ng mga mechanisms na to, yung pag-repair, dun na kapag hindi na kinaya, then that will lead to rising of a condition, no, mutation, okay, leading to disordered nga mga DNA repair capabilities. Okay, so halimbawa, we have xeroderma pigmentosum, we have Fanconi's anemia, ayan. Then we have also ataxia, telangiectasia. Okay, so just read the characteristics of these 3 diseases. Yung main concept lang dito is yung 3 diseases na to represented on the slide is that nagre-resulta ito sila, okay, dahil sa pagkakaroon ng disordered nga DNA repair capabilities. Okay? Hindi kinaya yung hindi kinaya yung pag-repair, okay, ng mga problem, mismatching halimbawa, yung base excision na nangyayari, hindi kinaya ng katawan natin to repair that. Okay? So, meron tayong mga arising disease like this. So, just read this one.
For lesson 3 of nucleic acids, we will discuss here all about gene expression. Particularly, we will be learning how this collective term gene expression relates to these terms transcription and translation. Now, first things first, what is a gene? Para malaman natin kung ano yung ibig sabihin ng collective term gene expression. Please take note that a gene that is only a portion or a region of a DNA. I repeat, a gene basically is a portion only of a DNA molecule, which particularly or specifically denotes to phenotype of an organism. When you say phenotype, that is all about the traits, the characteristics of an organism that describes the characteristics, the traits of an organism. So I repeat, when you say gene, that is just a region, a portion of a DNA that describes, that indicates, that denotes a phenotype, a characteristic or a trait of an organism. So halimbawa, this is a DNA. It is a double-stranded, di ba? Helix. Now, kapag sinabing gene, meron lang portion ng DNA, okay, that is considered a gene. So halimbawa, yung highlighted or boxed with color blue, yung portion na yan, okay, of a DNA, that is a gene. Okay? And a gene, it has to be expressed. Okay? It has to be expressed. And that expression, okay, in order to express, okay, the gene or that portion of the DNA, we have this process called transcription. Okay, so we have transcription, and this process of transcription expressed this gene into an RNA. So please take note, during transcription process, the gene serves as a template here. Okay, so kumbaga yung portion ng DNA, which is a gene, will serve as a template para i-transcribe to make it or to form, to synthesize RNA. Now, this RNA product from transcription will be further expressed. Okay, it will be further expressed, and that process is called translation. Okay, so we have translation process. Now, with regards to translation process, this involves expression of the RNA forming proteins. And proteins, again, this is represented by the elements carbon, hydrogen, oxygen, and nitrogen. So that represents a protein. So that's translation naman. The product in translation is protein. So I repeat, kapag sinabing gene expression, you have a gene, a portion of a DNA that is being expressed or being served, being used as a template. It will serve as a template to form the RNA. And that process is called transcription. And the RNA will be further be expressed, forming a protein via the process translation. So these are the two processes that will be discussed by Sir Chris. Okay, so I want you to access the video material on transcription and translation. It will be discussed in detail by Sir Chris from CAU Pharmacy.